I'm Evgeny Bondarenko, and I'm currently an assistant project scientist after doing a postdoc in Jack Feldman's lab. Jack Feldman is a world leader in respiratory neuroscience here at UCLA. He discovered the small area in the brainstem, which is the engine of breathing. And my whole idea was to try to use the expertise and the knowledge of working with this extremely tiny structure to design drugs that will selectively excite breathing. And we're doing that with the help of two chemists from UCLA and USC. Postdocs from their labs will join ReBreathe when needed. The first indication, the only indication I'm going to talk about today, is the opioid overdose. All opioid deaths are due to respiratory arrest. And opioid epidemic is a national emergency in the United States. It's the leading cause of death in adults from 18 to 45. The number of opioid fatalities has been increasing year over year and has recently reached the new all-time high. Opioids inhibit breathing by inhibiting the breathing engine located in the brainstem. Naloxone is the current only treatment for opioid-induced respiratory arrest. It blocks opioid receptors in order to restore breathing. However, since it's an opioid receptor antagonist, it cannot be used preventatively. It cannot be self-administered because it can only be administered when an overdose occurs. And it also induces extremely severe acute withdrawal symptoms. So our idea, instead of interfering with opioid signaling, is to excite the engine of breathing by other means, particularly by excitatory G protein coupled receptors. We see three primary applications in the opioid space. It is supplementing or formulating with medically prescribed opioids, such as opioids prescribed for analgesia or as part of chronic opioid replacement therapy. Alternatively, we can also supplement naloxone so that less of a naloxone is needed to overcome an overdose, and less naloxone means less of acute withdrawal symptoms. I found expression of two receptors for two peptides in the breathing engine on the same neurons that have opioid receptors. And when I pretreated animals with brain-wide infusion of agonists of these receptors, I found that it increased the lethal dose of fentanyl about 3 times. Additionally, as part of our R&D process, we will screen the breathing engine and determine other receptors expressed there, and will supplement our drug design process as needed. We have an NIH grant for that, so we're currently seeking funding for the drug design to perform an analogs of peptides, to create analogs of the 1 or 2 peptides that we have found to be effective for opioid overdose. I didn't have time to talk about other indications, but we believe that the same indication, the same drug can be used for central sleep apnea indication and for a couple of other respiratory conditions as well. Thank you.
So question around the phenotypic screening of those neurons, is that done ex vivo?
How I did that?
Well,
yeah. So I identified it with fluorescent in situ hybridization, but we also have in vivo data with. So this is in vivo through brain-wide infusion. We have animal, like cream mouse models of them as well. So
and how are you getting your readout?
With this?
Yes.
Oh, this is just an animal breathing. So I anesthetize the mouse, and this is actually very tricky because the awake mice, they don't overdose on opioids, and only mice that are anesthetized with a particular old school anesthetic, they do. And since our lab is old school, we have this anesthetic. and then I just infuse fentanyl and see how much they take until a full cessation of breathing. So I simplified the slides. Normally I show like the actual respiratory trace and how it changes from normal breathing to depressed breathing to a complete respiratory arrest as I infuse more fentanyl.
These are peptides. These are peptides that
Yeah.
are injected.
So these are peptides.
Mhm.
They have very limited distribution. The receptors have very limited distribution in the brain. They have some role in gastrin system, and one of them is also involved in itching. If we have too much of the side effects,
Sorry, involved in
Itching.
Oh,
itching,
okay, I see. Okay.
itching, yeah. So that's why they were never commercialized, because they have like no particularly great role in anything, and no one really tried to make a small molecule analog of them.
And these
Yeah.
are conserved receptors from, I
Sorry?
assume you're doing these in rats, the studies. So these are conserved receptors from rats to humans.
Yeah, yeah.
Okay, so
Yeah. So as part of um, the platform part, we will also check human postmortem tissue as well to make sure that they're expressed in humans.
sorry, to the itching point that you mentioned.
Yeah.
Do you suppress itching or they
No,
promote itching?
they actually promote itching. So
Okay.
the way around that is that they do it peripherally. So if we run into too much of a side effect, then we can combine the brain penetrant agonist with a peripherally restricted antagonist of that receptor.
But you get enough brain exposure that you
So it would cross the blood, only the agonist would cross the blood-brain barrier and act in the brain. And there are small molecule antagonists of those
I
receptors.
guess like the obvious question is you have to inject it versus the nasal spray. Like those are two.
Okay,
very
so
different.
naloxone is
Yeah.
very different there as well. It might not necessarily, we will have to inject. It depends on how, what kind of opioid we're supplementing or formulating. A lot of the medically prescribed opioids, they are taken orally, so
Yeah.
quite, they have like a slower onset, and therefore we will be able to supplement or formulate through oral route. For illegally used opioids, possibly, yeah, they might need to be injected or might need to be done. The market research, I did not base any of the market research on the illegal opioid field.
Yeah.
I've spoken with consultants. They said it's a fantastic market research problem. They would be happy to work with
I'm
it
sure they will be
because
happy
it's
to
so
do it.
interesting.
But I guess where I was going with this is you may not necessarily want to go with the acute use.
Mhm.
You may actually have a better use of it in the case
Yeah.
of say pain or other indications. So you know it could be interesting. Have you thought in those terms as well in terms of developing?
There are a lot of, as far as I understand your question, so there are a lot of legitimate uses for opioids. They're still the best analgesic that there is. They're still used in the hospitals a lot. And in the hospitals, for example, when they're used and breathing is suppressed, they have to intubate and mechanically ventilate a patient. If they can supplement it with something else that will, you don't need that. Then another use is, of course, for pain. So there are opioids like oxycodone is prescribed, and it's generally co-prescribed with naloxone. But people just keep naloxone at home or whenever they're taking oxycodone in case of an overdose, and will only use it when an overdose occurs. With this, it will be a continuous supplementation.
Can you talk a little bit about where you are in the IP process and expand on that?
In the R&D process, so we got the chemistry team together, and we're either trying to find private funds or applying for NIH grants to do that R&D process. the problem with NIH grants is that it takes a year until you get the money. So that's why we're exploring whether we can get private funds to do it much faster, essentially.
As far as your patent protection, is it a, assume it's a,
So what we have currently, it's not patentable because it's just an expression of receptors and the efficacy of naturally occurring peptides. I changed this actually this is the first presentation where I'm not specifying the receptors because the last one is a synthetic that I've tried, the synthetic analog, and that's where you actually start having some IP about with efficacy. The main IP will be the actual chemistry part.
So the chemical matter is peptides.
The chemical matter is peptide.
Why peptides and not small molecules?
No. Right now, those are peptides. So we will need to create analogs of those peptides. They just don't exist yet. But they are extremely great targets to go after and have many, many good uses.
Talk about the use of funds and how it's creating value in the process, and also maybe more about your platform too.
All right, so yeah.
First question is use of funds. If you talk about how is the use of funds creating value? What are the value inflection points that you're going to hit with use of funds? Is it all to advance these candidates that are attacking these two targets that you mentioned?
So
and then your platform, if you could share more about the platform too.
All right. So the use of funds, it will be for drug design, for the chemistry part, where we will do crystal structure, and then the actual drug design of analogs, and then testing them in a GPCR binding assay specifically designed for the GPCRs, test the blood-brain barrier permeability, heart and lung toxicity, and then the efficacy in vivo. At the same time, we have the NIH grants now to do a scale up of the platform where we will identify other potential targets expressed in the engine of breathing, test the efficacy in vivo with peptides or whatever, whichever way I can stimulate them, confirm expression in human postmortem tissue, and also investigate side effects. Yeah. All right. Thank you.